I'll be speaking about the results, these are the partial results, from the NTP chronic carcinogenicity study that I mentioned previously began thinking about in the year 2000. Okay. Within the U.S. government, there are two agencies that are particularly involved in RF radiation and health effects. One is the U.S. FDA, and they nominated cell phone radiation to the NTP. And the reason for this was back in the year 2000, exposure was widespread, but nothing like it is today. Little was known about the potential health effects from long-term exposure. There had been some studies, but they were of not decent quality. The guidelines then were based on protection from acute injury from thermal effects. Epidemiology studies that were available at that time, and there were some, indicated a potential increase in glial cell tumors in the brain, gliomas, and vestibular schwannomas. These are acoustic neuromas. A large part of this was based on work done in Sweden by Hardell, and they were considered maybe associated with cell phone use. However, there were a number of inconsistent results in the literature, and studies in laboratory animals did not show an association with exposure to RF. And in the year 2011, the International Agency for Research on Cancer, which is part of the WHO, classified radiofrequency radiation as possibly carcinogenic to humans. Okay, so as I mentioned, in 2011, IARC determined that there was cell phone radiofrequency radiation was a possible human carcinogen. This was based on what's called limited evidence in human. There had been positive associations observed with exposure to RF radiation from wireless phones. These were the gliomas and acoustic neuromas. There were also negative cohort studies. However, these have the potential for misclassifications when you're looking at large populations and you don't have exposure information on the individual. The positive case control studies, which showed the gliomas and acoustic neuromas, are considered to have a potential selection bias. Therefore, the evidence was considered limited, not sufficient by IARC. The evidence in experimental animals was also considered limited because the whole animal studies were negative. There were some positive studies with co-carcinogenicity. Overall, then, the evidence was considered possibly carcinogenic to humans and group 2B. So what does limited evidence mean? Limited evidence means that a causal interpretation between exposure and cancer is credible, but chance and bias or confounding cannot be reasonably ruled out. Limited evidence in experimental animals indicates that a carcinogenic effect, but not necessarily definitive because it may be only a single study or benign neoplasms, was observed. So possibly carcinogenic to humans indicates that there's limited evidence in humans and less than sufficient evidence in animals. To reach a level of probable human carcinogenicity to humans, limited evidence in humans would be adequate if there is sufficient evidence in animals. And this is an important point in terms of the use of the animal studies. So why do we use animals for evaluating human cancer risk? As mentioned before, there's similar biological processes of disease induction. It's unethical to intentionally expose humans to test for carcinogenicity. It may happen, but it's not intentionally exposure. Every known human carcinogen that has been tested adequately is carcinogenic in animals, and about a third of the human carcinogens were first identified in animals. Animal studies have also a very controlled exposure. They eliminate any potential confounding. And animal studies can eliminate the need to wait for very high incidences of a long latency human cancers before implementing any public health protective strategies. So the agencies in the U.S. that are concerned about radiofrequency radiation are the FCC. They established the exposure guidelines. This was done in 1996. The FDA, who nominated cell phone radiation to the NTP, is also involved, they would be performing a risk assessment on the data from the NTP. Within the U.S. government, the data may be prepared or developed by agencies such as the National Toxicology Program, but it's the regulatory agencies that do the quantitative risk assessment. Other agencies interested include National Cancer Institute, Environmental Protection Agency, and National Institute for Occupational Safety and Health. So animal studies that had been done around the year 2000 and studies that were done in the early 2000s used what's considered a Ferris wheel structure like this. Animals are placed in tubes in this Ferris wheel structure with the antenna through the center. So what's happening as this moves around, the animals are exposed, and because they're in tubes, the exposure duration per day is 2 hours. But as you can see, one side of the animal is exposed while the other side is further away from the antenna, but then it receives its exposure on the other side, like moving in a Ferris wheel. Therefore, the actual exposure to tissues is really 1 hour per day. And in terms of a toxicity study, what we want to know is what is the potential risk to humans from animal studies. So how do we typically do this in a toxicity study is we increase the dose so that because our sensitivity is in the range of 5%, which is an extremely high level if this was a rate occurring in human populations. Increasing the dose is one way to demonstrate whether there is an effect and use a quantitative risk assessment to evaluate risks in the general population, which may be of the order of 1 in 100,000. Obviously, you can't get 1 in 100,000 risk from an animal study when group size is 50 or 100. So our interest was, how do we increase exposure, but we can't increase it too much because of the thermal effects. So what we wanted to do was to increase the time of exposure. A 1 hour or 2 hours exposure we considered to be very limited. And it was because of this that the NTP began to explore alternative approaches to exposing animals. We established a collaboration with the National Institute of Standards and Technology in Colorado and with ITIS in Zurich to develop an exposure system to conduct animal studies where we could expose them for almost a complete day, because we knew our exposure dosimetry would be limited by the heating effects. We used 900 and 1900 megahertz because these were in the range of carrier waves used in telecommunication systems. Okay, at ITIS, the chambers were built they were based on a prototype chamber. We demonstrated the technical parameters for exposure of animals to show that in a reverberation chamber there would be equal electromagnetic fields in all regions. The 21 chambers were constructed in Switzerland. They were shipped to Chicago, where the studies were actually conducted. We used 2 signal modulations, the ones that were most used in the US and Europe at the time, CDMA, code division multiple access, and GSM. So because we had 3 exposure groups and a control for each male rat, male mouse, and female mouse, we had 21 total chambers. For rats, males were in separate chambers from female rats because of the larger size of a rat compared to a mouse in terms of allowing exposure within a chamber. These chambers are about 3 meters by 4 meters in dimension. They're essentially large microwave ovens. They were installed in Chicago. Here's a chamber being loaded down, brought into the basement. This is what a typical chamber looks like at the door. It's this is the row of 21 chambers in the Chicago facility. Now, what is a chamber? As you can see in the far left over there, where we have an antenna and a paddle, The antenna is where the exposure is emitting from. The paddle distributes the electromagnetic fields throughout the chamber, and through probe measurements we're able to demonstrate that within the cages of every cage they were statistically comparable in all regions. So this is a chamber with 2 cage racks. So this cage essentially had the same electromagnetic fields distributed throughout the chamber. The issue that's most important on reverberation chambers is that the fields emanate from multiple angles. These are coming in from all directions. So it's not just one antenna directly at the animal. The fields are distributing throughout the chambers, coming in from multiple directions, creating that statistically homogeneous electromagnetic environment. Based on using this kind of a system, then, we would not have a limit on exposure time, and we could expose animals as long as necessary. We figured 18 hours with a 10-minute on, 10-minute off would be sufficient to challenge the null hypothesis that there are no effects associated with cell phone radiation. Okay. This is data that was presented to us from ITIS. They developed mouse and rat models, and as I mentioned, the fields are coming in from all directions. This is one in which they calculated the relative absorption in tissues when you have 12 different polarizations. And we looked at the mouse at 900, the mouse at 1.9 gigahertz, the rat, and you can see that in the mouse at 900, the tail is the strong absorber, and in the rat at 1900 megahertz, the tail is the strong absorber. Obviously, we're not interested in just exposing the tail of the animals and excluding the other tissues. That was the reason why 900 megahertz was used for the rats and 1900 megahertz was used for the mice. The absorption is less in the tail, and you can see that our main interest was in the brain, where there's very little deviation. So that when we talk about a SAR, specific absorption rate, of 1 gram per kilogram, watt per kilogram, the level in the brain is essentially the same as that which is presented to the whole animal. So this is showing a deviation. Obviously in the fat, which is a poor absorber, the dosimetry in the fat tissue is less compared to the relative whole body, because it's fat tissue which does not absorb. So the NTP studies were conducted in 3 phases. One was a thermal pilot study to determine what would be the exposure intensity that we could use without overheating the animals. That study was conducted at 4 and 12 watts per kilogram. We then conducted a 28-day study to determine would there be effects that might be detrimental to a study that would be conducted for 2 years. Then we conducted the 2-year study. In all studies, the exposures were essentially 9 hours per day, because the total was 18 hours and 20 minutes with a 10-minute on, 10-minute off cycle. So that when one chamber was exposed, a different chamber was unexposed, and we could use amplifiers for 2 chambers throughout the daily exposures. Okay. So rats were exposed to both GSM and CDMA at 900 megahertz, and mice were exposed to GSM and CDMA modulated signals at 1900. Quickly, this is a demonstration of the thermal effects associated with exposures by increasing the SAR. We used animals of different weights, different ages. This was done in rats and mice. We used pregnant rats, and you can see that our interest was not to allow a 1 degree temperature rise in centigrade. And at 12 is obviously too high for males, but in the region of 6 watts per kilogram, or less than 1 degree rise in whole body temperature. These were determined with microchips implanted in the interscapular region of the animals. So these results suggested that the 6 watt per kilogram would be adequate for a chronic study. In the 28-day study, rats in our studies began exposure at gestation day 6. So these are in utero exposures. They're exposed from gestation day 6 through lactation and then 2 years post-lactation. There was increased pup loss when went to higher exposure doses. Okay. In May of this past year, NTP released the partial report. And I call this partial because not all of the data have been reported, but all of the incidence values for brain tumors and schwannomas have been completed. The NTP goes through a 3-phase period of pathology review. It's first the original pathologist from the study laboratory, reviews 40 tissues per animal. There's nearly 3,000 animals in this study. So the pathologists from the laboratory make their first determination. It then goes through a quality assessment review by another pathologist. They read all of the identified lesions, especially the tumor lesions, as well as 10% of unidentified animals with no lesions to see if anything was missed. And thirdly, it goes through what's called a pathology working group, which is a group of pathologists, not just from NTP, but group of pathologists, including NTP pathologists and outside pathologists with expertise in the areas of interest in brain or heart. These people then review the differences between the quality assessment pathologist, the original pathologist, and come up with the final determination of what the incidence values are. That has been done, and that will not be repeated because these then become the final numbers. Okay. So why did the NTP release this information in a partial format? Well, many reasons. There's much public interest in this kind of findings. Because of the widespread use of cell phones. The tumor types that were identified here were gliomas and schwannomas of the heart, which are similar to those that were observed in the epidemiology studies of cell phone users. Therefore, these findings then support the IARC conclusions of a potential carcinogen. And when we look at incidence values, you've got to remember that there's a widespread global use of cell phones. We're talking in the multi-billions of people who use cell phones. So that even if there's a small increase in incidence, the issue is really what is the risk? Because the incidence is incidence in animals of group size 100. We can't distinguish if it's much less than 5%. But through a risk assessment approach, you can evaluate what would be the risk with exposures, different exposure durations on the risk for cancers that are identified within this study. Okay. The NTP used Harlan-Sprague-Dawley rats. The SARs were 0, 1.5, 3, and 6. GASM, CDMA modulated in utero on gestation day, beginning on gestation day 5. There was an interim evaluation at 19 weeks, and the study terminated after 2 years. Okay. Within the perinatal effects, there were no exposure-related effects that were observed in this study on pregnancy or littering, but there were decreases in body weights of male and female pups on postnatal day 1. So animals that were exposed in utero were of less body weight, the pups, when they were born. Also, after lactation for 3 weeks, there was the body weights of the young animals was still less than the control animals. Now, survival of rats in this study is an important issue because the survival appeared to be better in the treated than in the controls. The filled-in squares are the controls, and this is the unfilled squares are the, I'm showing here the CDMA 6 watt per kilogram. This is the highest exposure, and you can see that during the course of the 2-year study, the CDMA actually had poorer survival. Once we got out to week 95, they were essentially the same, and then control rats were dying at a faster rate than the CDMA. Why that happened, I don't know, but this is an issue that has been raised a number of times, so we've got a question, were the animals at risk, were they at sufficient risk to develop tumors? You can see that if by week 95 there's no difference, in fact, the statistical comparison between these two is non-significant, which leads me to feel that this was not a non-issue within the CDMA experiment. Okay. So currently, the only findings that are being reported are the identified target organs. The two were in the heart, which were schwannomas. These are of the nerve sheath, and gliomas. This is a histopathologic diagram of the heart, which identified the schwannoma. I'm not a pathologist, so I can't see the cell type clear enough. It's pretty clear to a pathologist, but it's in this particular region here. I can't see the cell, what the cell nature is, but the staining is obviously quite different. So in the heart lesions, there were proliferative heart lesions. The schwannomas, you can see in the GSM went up to 5.5%, with the CDMA modulation up to 6.6%, with none in the controls. There were also hyperplasias, Schwann cell hyperplasias in the heart. Hyperplasias are essentially a preneoplastic lesion. These are excessive growth, number of cells in a focalized lesion, which has that potential to go on to develop into a tumor. So if we look at all of these, you can see that with 0 in the control and in the CDMA modulation with the male rats at 6 watts per kilogram, we're approaching 10% of proliferative lesions in that study group. This is...
Could you say a word about why the controls in this study are so important as opposed to others?
Okay. Typically, you compare control animals to other studies in which there were control animals. However, in this particular case, we don't have a comparable control because in this case, the animals were exposed in these shielded rooms such that there were no electromagnetic fields penetrating from outside and nothing penetrating outside. The EMFs that were generated within were absorbed by the animals, and that's how we set the dosimetry. So we actually have no comparable control. When you look at historical controls, even among other types of studies, and inhalation studies are considered different than a feeding study, so that it's appropriate to compare historical controls by the nature of the exposure route, because in this case, it's very different within the exposure chambers. This I already did. Okay, this is a photomicrograph of a brain where the glioma is seen here, and the pathologist says it's very good. So I take her word for it. But you can see the differential staining within the site where the glioma is present. So of the proliferative lesions in the male rats, there were increases and positive trends for gliomas in male rats with GSM and with CDMA. And there's also the hyperplastic lesions, again, preneoplastic lesions that are observed in the exposed animals, none in the controls. So again, similar to the heart, we're up to about 5% of animals showing proliferative lesions. with both CDMA modulation and with GSM modulation. In female rats, there were incidences of gliomas and glial cell hyperplasias of the brain. The numbers were smaller than in male rats, but the controls were 0. And again, this was not significant, no significance, but it's hard to say whether these are biologically significant because of the observation, what was seen in male rats. Female rats tend to be less prone to developing brain lesions than male rats. So in summary, body weights at birth and throughout lactation in rat pups tended to be lower than controls. There were increased incidence of schwannomas in the hearts of male rats. There were significant SAR-dependent positive trends for both GSM and CDMA, and a significant increase at 6 watts per kilogram with the CDMA modulation. There were significant SAR-dependent trends for increased gliomas in the brain of rats exposed to CDMA-modulated RFR. The lesions in female rats were not significantly different. The conclusion in that partial report was that the lesions, the hyperplasias and glial cell neoplasms, were likely the result of whole body exposure to GSM and or CDMA modulated RFR. And the values seem to be more obvious with the heart lesions than in the brain. Exposures in female rats were non-significant. NTP anticipates that the complete data set for rats, mice will be peer-reviewed at a public meeting either the end of this year or early 2018. Lastly, within this study, as I mentioned, there was an interim sacrifice at 19 weeks, which is fairly early. It's not a chronic. And studies were conducted using the COMET assay to look for DNA strand breaks. And this was observed in male rats, female rats, male mice, and female mice. Here's observation for male rats of the frontal cortex as a function of the SAR. And this is a summary of all of the data where DNA strand breaks were observed in rats, male or female. The strongest responses were in the frontal cortex in both rats and mice. The trends were significant, and the pairwise comparison between the dose group and the controls were significant. DNA strand breaks were also seen in the cerebellum, the hippocampus, in males in the liver and in the blood, that would be peripheral lymphocytes, and the others shown here. So what the importance of these results? These results are quite important because the incidence of the brain tumors and the schwannomas of the heart and the exposure-related increases in DNA damage in brain cells support that IR conclusion based on gliomas and acoustic neuromas among long-term cell phone users. The exposure intensities were limited by the heating effect at higher levels. So we're limited in terms of how high an exposure we could go. And these are similar to or slightly higher than the RF emissions from cell phones. Cell phones in the US, the limit is established at 1.6 watts per kilogram. We used 1.5, 3, and 6. So we're not that different compared to cell phone emissions. As I mentioned, survival was sufficient to detect precancerous lesions in the brain and heart of controls. There was no statistical difference that I tried to show between male rats and the exposure group with the highest rate of gliomas and heart schwannomas. And there were no hyperplasias, glial cell hyperplasias, which are a precancerous lesion, or heart schwannomas in any control rat, even though there were glial cell hyperplasias detected in exposed rats as early as week 58. At week 58, there was no difference in survival between the controls and the exposed groups. So that if you see a preneoplastic lesion at 58 in a exposed animal, there was sufficient time for this to occur in a control rat as well. And in a 2-year study, there was a heart schwannoma detected as early as week 70 in exposed rats. So I conclude that the survival issue, which has been raised a number of times, is not really a driving force for the results that are observed in this study. NTP is looking into possibility of some follow-up work in terms of using smaller chambers to answer more of a mechanistic type of studies. Some of these relate to understanding oxidative stress, replicating some of the genetic toxicity findings, and maybe looking for molecular changes in the heart. And with that, I want to mention that the NTP study director now is Michael Wyde. He took over after I retired in 2009, and NTP associate director is John Bucher. The agencies that we worked with, NIEHS is part of the National Institutes of Health. The studies were conducted in Chicago at ITRI. NIST helped us, the physicists, in designing the exposure system, and the ITIS Foundation in Zurich was instrumental in working out the dosimetry, building the chambers, and validating the exposure intensities within them. Thank you.
Ron, I want to thank you again very much, because this was not what you had planned to do, and I think as the former study director and really architect of the study, we're very fortunate, very, very fortunate to have you here, and I really want to thank you.
Thank you.
Why don't we take questions now, and then we'll have a short coffee break. Tony?
I have two questions. I have two questions. The first is, do you have any data other than you've shown in relation to mice? Because they were at a higher level of exposure, as I understand it. And the second is, could you comment on the fact that in humans we're talking about acoustic neuromas, schwannomas around the acoustic nerve, whereas here you're talking about the heart.
Right. Okay. Well, the mouse study results haven't been reported. They were the same. The same SAR values were used for mice, the 6.3 and 1.5. One difference between them, well, there's two differences. One is the frequencies were different. Whether that matters or not, I don't know. But also, in the rats, I indicated that the exposures began on gestation day 6. In the mice, the intention was to expose them during exposure, but the B6C3F1 mouse that is used in NTP studies tend to eat their pups. So we had to not start them from in utero exposure. They began in the typical NTP fashion of at 6 weeks of age. And in terms of the acoustic neuroma versus schwannoma of the heart, yeah, they're obviously different locations, but it's the same cell type, Schwann cells. And that's why that concordance is one which lends to believe that these show that type of relationship that adds interest to the acoustic neuromas story.
Thank you very much for this. For this important talk and very clear talk. I think that this is really one of the most important studies that emerged on. Sorry? Yes. Sigal Sadetsky. But since it's so important, I want to, and thank you very much specifically for clarifying the survival issue, which wasn't clear up to now. If you could clarify a few issues. The first one is, if the incidence is so low among females, why did you include females in the first place?
Yep.
The second...
We didn't know that.
But don't you know that naturally female rats don't have lower rates?
We know that, but that's not a reason to exclude them.
Depends on the power calculation. This is what I'm asking.
Yeah,
if
yeah.
you know
Well,
in advance that you're not that the power calculations that you would not expect increased risk among females, why did you include them? This is number one. Number two is, do you think, according to these results, wait a minute. Number two is, I just want to make sure that the controls are right. And therefore, I would like to ask you, were you surprised that there were 0 tumors among the controls? Meaning, did you expect any background tumors among the controls? So these are two methodological issues. And my third question is, according to these, or based on these results, do you think that the IR classification should be changed to 2A or to 1, meaning definite carcinogen or probable carcinogen?
Okay, well, with respect to the female rats, as I mentioned, we don't know the results of the study until we conduct it, obviously. We didn't know whether we would see any tumors in male rats. Well, we don't know whether there would be an effect of RF on male rats or female rats. No, you asked me male, female,
Wait.
the male, female.
Hold on. I think what Ron is trying to say is that whenever you start out a study, you don't know what the results are going to be. There could be a possibility that even though the background would be low, the study could be causing this. There are examples, as you know very well, that you get breast tumors in male animals sometimes, even though you don't expect them.
No,
Right.
this is
Yeah.
what I'm asking you.
What-
Did you
what-
have expectations about the control? That's the whole thing
Yeah.
of control. You have an expectation
Yeah.
for the control.
The control, right, but it depends. First of all, we typically did not use Harlan spray dolly rats in NTP studies. This was the first study that actually used Harlan spray dollies. It used to be the Fisher 344 rat.
Right.
Switched over to a different rat strain. In terms of the control rate, as I mentioned, these were exposed in reverberation chambers. There's no external electromagnetic fields that these animals are exposed to. I have no idea what that might be doing to animals. Our interest, as we designed the study, was to make sure that the brain was exposed, because the brain was the focus from the epidemiology studies. That's why when I showed the graph of the different organ exposures, our interest was the brain, obviously not the tail, and the brain exposures were within 1 decibel of the whole body exposure. Secondly, because of the low rate of tumors that are seen in the brain of animals, we increased the group size. Typically an NTP study is done with 50 animals. We increased it to 50 to try to increase the power. Our statisticians told us to increase it further, we might not see a whole lot more power within the study. We might have to go to a very large number, and as you can see, we could only fit 100 animals in rats in an exposure chamber. We would have had to double, triple the size to increase the power even further, where we might have seen a control rat with a tumor. But
Perhaps you can
these are
talk
the
more
data.
about this at the coffee break, but go on to the other
I forgot the
questions.
third point.
The question was about whether you think it should be reclassified in light of this. Right,
This will not make it a known human carcinogen without to be a known human carcinogen by IR classifications would require sufficient evidence in humans or strong animal data with strong mechanistic data. And part of the discussion, I think, at this conference will be related to the mechanistic data. How strong is it? As Deborah was talking, the issues related to oxidative stress seem to be showing up quite fairly consistently among studies, and this may be leading into a mechanism that is one of the characteristics identified by IARC in a paper by Smith last year, characteristics of human carcinogens. One of the 9 or 10 characteristics includes oxidative stress.
So
and genetic damage.
Is it your personal opinion that it should be that you were a member of the IARC monograph?
I was.
Okay. So if you were presented with these data now, would you continue to say it's a possible or a probable?
No, I would say it's a probable human carcinogen.
Right. So it would be a 2B. All right. We have lots of questions here, so I think I'm going to try to limit the questions to very, very brief questions. Okay, Frank, you have one.
So I'm sure it's very hard to pathologically look at acoustic nerves in rats. But I wonder whether there may have been schwannomas at other sites, because the highest frequency in humans would be in the brachial plexus, the lumbar plexus, and the cranial nerves. So I doubt that those areas were examined pathologically, because they're hard to find and hard to examine. But it might be interesting in the future in similar studies to do animal imaging and actually look at those regions.
Okay. The NTP cuts multiple sections in the brain, but I can't address your question. Maybe someone else can.
If you want, I can explain you during the coffee break, because the head is completely decalcified and then sectioned. We do in 5 parts, I think you 3 or 4 as MTP, and then all the sections are examined, and you can believe that, but it is quite easy to find the acoustic nerve in rats. It's more difficult in mice, but with rats it's easy.
Regarding the same topic, cardiac schwannoma is extremely rare in humans. There have been only 16 case reports in humans in the literature.
Okay.
So now regarding the origin, the neural origin, was it the vagal nerve or the cardiac plexus, the sympathetic or the parasympathetic innervation of the nerve of the heart?
We'll have to
That-
ask.
I don't know.
But we can get the answer to that, and we'll note that question, Serena. Was it the vagal nerve or the sympathetic nerve? We'll ask our colleagues, and we'll get an answer to that question.
I don't know the answer to that. Sorry.
I have 2 comments about NTP study and 1 correction comment to Devra's study, Devra's presentation. About this NTP study and significance of this rat data, this brain cancer, I go further than, as Sigel mentioned, when you don't have any cancer in controls, then automatically when you calculate these few cancers, 1 to 3 cancers per 90 animals in exposed groups becomes more significant. So because of this absolute lack of cancer in control group, your results become more significant. The other comment is this, that when you speak about DNA damage, you don't exactly know whether this RF caused damage that you detect with RF, with this COMET assay, or not. You don't know this, because you don't know whether RF damaged DNA or whether there was spontaneous damage of DNA which occurs normally, and RF inhibited repair of this. But when you have no micronuclei at all, then it is very difficult or impossible to say that there is any indication of genotoxicity.
Okay.
And then
I don't
correction.
know the mechanism by which the strand breaks were detected, whether it was an induction of the strand breaks or inhibition of repair.
These
Obviously,
are good questions that I think that we will pass on to the colleagues at the NTP. And then you had a correction of something I said.
Yes, namely, in this Agarwal review, from which you have taken image of this mechanism, where we have these different stress pathways in sperm. This is fake. Because he is taking HSP27 data directly from our endothelial cell study. He is saying that HSP27 is activated, phosphorylated in sperm, which leads to activation of apoptosis in sperm and activation of blood-sperm barrier in humans. This is not. This HSP27 data was generated in endothelial cells concerning endothelial cell apoptosis and concerning blood-brain barrier. Study on HSP27 in sperm was not done by Agarwal. We have done HSP27 study in sperm with my student in South Africa. No activation whatsoever.
Okay, that's your one study, but in fact, other studies have looked at heat shock protein 27 in sperm, not in Agarwal, and we can talk about that at the coffee break. I will show you some of the other references. All right. I understand the reason why we're having this meeting is because there are debates in science, and I think it's really important, and I want to
Me too.
thank you all because I know
I want to
we
I
can. We
have an
can-
important comment
Okay.
to Darius. So when you question on the genetic effect, whether it was a direct effect of the RF or an inhibition of repair, in either case, it is showing that in the exposed animals, there is a biological effect that is occurring. Whether if it was induction by RF, it might be through oxidative stress and reactive oxygen species, whether it's inhibiting. But in either case, it's showing clearly that at 19 weeks, there is in rats and mice effects on DNA structure that are caused by that exposure. How it's happening, I can't tell you,
Right.
but there is an effect, and that's
And-
causal.
Right. And I would just like to end the conversation right now. We will resume. We'll have more questions after Fiorella's talk. But in the interest of our getting coffee, I want to thank you all. I want to say